we're going to move it over here, and we're going to take some carbon dioxide and 3 of these. Do you guys remember what that 5-carbon molecule is called? RUBP. Or Rubisco. That is RUBP. That's wrong. That's the wrong molecule. It's RUBP. So Rubisco. So what we're going to do is we're going to take 3 of these, and we're going to attach to it 3 carbon dioxides. We're going to just draw those as a single circle, right? So that we get a very short-lived 6-carbon molecule. And these are the dark reactions. This is occurring in the absence of light. We don't need light for these to occur. What do we call that phase?
Calvin cycle?
So it's all part of the Calvin cycle. Fixation. Yeah, the Calvin cycle consists of 3 discrete phases, the first of which is fixation. And fixing just means that you're taking one thing and sticking it onto another. So we're fixing carbon dioxide on some Rubisco. And we created a 6-carbon intermediate. So total, how many carbons do we have in Rubisco? 15, exactly right. So we have 15 carbons here. And how many carbons total do we have here in our carbon dioxides? How many carbons does each carbon dioxide have? Right, so carbon dioxide looks like this. Carbon, double bonded to an oxygen, double bonded to an oxygen. So, yeah, there are 3 total carbons right here, right? Because each carbon dioxide molecule carries just a single carbon. So we have 3 carbons here. Now let's think about this for just a minute, you guys. What we're doing is we're taking 3 carbon molecules and we're bringing them into the dark reactions. And ultimately what we want to do is fix those 3 carbon molecules into a sugar, okay? And the way that we're doing that is by using some carrier molecules, our RUBP. Okay, so RUBP comes along and it picks up those carbon dioxides. So we get 3 of these 6-carbon intermediates. So how many carbons do we have represented right here?
18.
18, exactly right. So what we're going to do is we're going to take those 3 6-carbon molecules and we're going to invest some ATP and some NADPH. And we're going to make them into 6 3-carbon molecules. So we take some ATP and we convert that into ADP. How many, do you guys remember how many ATPs we use at this point? 6, exactly right. We get 6 ADPs. What that means is each one of these 3 carbon molecules actually has a phosphate attached to it like this. Those are all phosphates. And how many NADPHs did we use?
6.
Also 6 of those. And those are just dropping off electrons, right? Do you guys remember what the name of this molecule is? This 3-carbon intermediate? Yeah, it's G3P, glyceraldehyde 3-phosphate. it's glyceraldehyde 3-phosphate, and those phosphates are coming from our ATP molecules. So what did we call this phase right here?
Reduction.
Reduction. Yep, this is the reduction phase. What don't we like about this reaction so far? What's confusing about it? Because there's definitely some angst in the audience today. What is it that you guys don't like about it?
It's not free flowing like that one.
Okay. Do you understand why we're counting carbons?
No.
No. Okay, so the reason that we're counting carbons is we have two reasons, right? We need to know exactly how much sugar we're making. And sugar just consists of a bunch of carbons, hydrogens, and oxygens, right? And for each glucose molecule, how many carbons are there? 6. Okay, so that's an important thing to keep in mind. The other important thing about this reaction is, because this is an enzyme, we have to be able to reclaim it. We have to remake it in some way. And so we need to know how many carbons from this reaction we need to hold on to so that we can make back our Rubisco. Now, how many carbons were there in Rubisco? 15. And how many carbons are there right here? 18 carbons right here. So if we want to make back our Rubisco, how many of those carbons do we need to hold on to? 15. And how many G3Ps is that? It's 5. So we have to hold on to, these would be rad, right? If we could just take all of those and make them into sugars. Because just from those 6 G3Ps, we could make how many sugar molecules? It's close to 2. 1. It's a little bit farther from 1. 3, yeah. We could make 3 of them, right? Because we can have 1 sugar molecule here, 6 carbons, 1 sugar molecule here, 6 carbons, and 1 sugar molecule there, 6 carbons. So we could make, potentially, 3 sugar molecules just from this one reaction. However, that would leave us without any Rubisco. So we have to take enough carbons from this reaction to make our Rubisco back. That's why this carbon accounting is so important. This truly is an enzyme, you guys. It has to be reusable. Yeah, Adam.
So it has to go through that cycle twice to make it sugar.
Yeah, it does. Exactly right. So before we can get there, though, we have to make back our Rubisco so that we can go through it again, right? So we're going to take those 5 G3Ps and we're going to make them back into Rubisco. And that's going to require a little bit more ATP. And how many ATPs did that require? 3. So we invest a little bit more energy to make back our Rubisco. And that was called what? Reclamation. That's a name I made up for it. It's not actually what it's called. That's just what I like to call it. It's actually called the reconstruction of of Rubisco, which doesn't sound nearly as cool as reclamation, right? I mean, reclamation sounds like what we went through after the Civil War or something. Okay, so that's the dark reactions. And you guys, the dark reactions, they're all occurring within the thylakoid. So all of the stuff that we're making in photosynthesis, in the light reactions, stays within the thylakoid, and we use the proteins within the thylakoid to make G3P. The actual product, you guys, the actual product from photosynthesis is not sugar, it's not glucose, it's G3P. There's a whole other series of reactions that we're not going to talk about that takes G3P and makes glucose from it. So on the exam, when I ask you, what's the actual product of the dark reactions? I'm going to have glucose as an option, and I'm going to have G3P as an option, and y'all are going to say G3P. It's G3P.
Wait, can you repeat that?
Yeah, you bet. So on the exam, when I ask you what the actual product of photosynthesis or the dark reactions is, it's G3P. It's not glucose.
All right.
Okay, so does this kind of make sense?
Makes complete sense.
I'm glad. Yeah, Carly.
So on the G3P, is that just a phosphate that's attached to them?
Those orange things?
Yes.
Yeah, those are just phosphates, and they're all coming from
From
our
the
ATP molecules. Okay, so now that all of the phones are down, let me just remind you that it's my preference that you not email or text in class, right? So if you need to answer a text or something, I don't mind if you just pop out. Okay, so questions about, yeah,
I have a question.
Sarai,
I'm
fire away.
kind of confused with the ATP and the ADP part. Like, I don't understand what that means.
Good question. So what's the relationship between ATP and ADP? So let's remember what, okay if I erase this? Flip over here. Let's remember what ATP actually is. So ATP looks like a nucleotide, right? It's just an adenosine nitrogenous base attached to a sugar that's attached to some number of phosphates. And so it looks like this. There is a ribose.
This is ATP.
This is ATP. And this ribose is attached to a nitrogenous base called adenosine. Yeah, so this is, I'm just going to put a capital A in there. So we could take this, so far, if it had a phosphate attached to it, it makes some RNA out of it. It's just a nucleotide, is all it is. What makes it ATP, and A comes from the adenosine, right, is coming off of the ribose on the other side. There is a phosphate. So that's our whole nucleotide, right? It's a phosphate attached to a ribose and a nitrogenous base. But instead of just having one phosphate, we have 3 phosphates. Like that. And I put a wavy line right here because this is very unstable and super high energy. Why is it unstable and super high energy? Why not? Yeah, so the phosphates don't like being by each other because they're all negative. And sticking 3 negatively charged molecules together, it's unhappy. So this is ATP. It's this stuff attached to 3 phosphates. ADP is this stuff attached to 2 phosphates. So the way that we use ATP for energy is we take this terminal phosphate and we drop that phosphate off on something else, and that energizes it. It's like sticking a battery on it. So every time we use an ATP, that ATP, adenosine triphosphate, loses one of its phosphates and becomes adenosine diphosphate, which is a much more stable molecule.
Since you're using that to get down those phosphates down to that step with the 18 carbons, why even have the NADPH?
So the NADPH, I haven't shown you all the side groups on those. These molecules are actually relatively complicated. They have oxygens and hydrogens coming off in really odd places. And sticking those things together requires some electrons to complete the covalent bonds. And the source of those electrons is our NADPH. Okay, so right, does that make sense?
Yeah.
Okay.
So that diagram right there, that's ATP.
This is ATP.
Okay.
Yeah, and if I was to just chop off that terminal phosphate, that would be ADP, adenosine diphosphate, for 2 phosphates.
So that's ADP like
This
that?
is ADP. Yeah,
So
Carly.
the conversion of ATP to ADP provides the energy, then the NADPH conversion to NADP plus is kind of like the stuff that sticks together.
Right, it's the electrons that are helping make those covalent bonds. And what happens to our ADP and our NADP plus? Where do these things go. They just go back to the light reactions, right? Pick up more phosphates and more electrons. Yep.
Oh.
Sophia.
Sorry to stretch, and I have a question. So for the NADP plus, why is it a plus if it's like, if that's a negative?
Wonderful question. So remember, these two molecules, ATP and NADP plus, are different. This is just energy, and this is something totally different. NADP plus has that plus on it because it actually carries a positive charge. And that positive charge allows it to pick up electrons at this stage. But it's not just picking up electrons, it's actually picking up some electrons and some protons. What was that, Carly?
Oh, I was just thinking, so that reduces the positive charge because you're adding another electron.
Right, which is why this is neutral. Yeah,
Oh, okay.
good question. Others? Okay. Okay, so this is photosynthesis in all of its glory. There's one more thing that I wanted to talk to you about in the context of photosynthesis, and that was something called photorespiration. Now, gas exchange can occur in plants because they have these little pores called stomata. And we talked a little bit about them at the beginning of chapter 6. They're pores that are typically on the underside of the leaves that allow carbon dioxide to come in and oxygen to go back out, right? You know that oxygen is created during the light reactions. However, it's not just gas that they exchange with the atmosphere. It's also water. So if plants get too hot, they become dehydrated. They lose water. They start losing water through their stomata. So the plant's natural reaction to that is to close the stomata so that they don't become dehydrated. But when you close your stomata, you can no longer exchange gas either. And because it's still during the daytime, the light reactions continue to occur. Because the light reactions are continuing to occur, oxygen gas, which is a metabolic poison, builds up inside the plant's leaves. And if it continues, it could actually kill the plant. Now, what's interesting about the Rubisco molecule is it's equally sensitive to both oxygen gas and carbon dioxide. So